Hi, and welcome to Chapter 11, Bio 101. We are going to talk all about genetics today. And specifically, we're going to talk about the kind of genetics where you're passing characteristics and traits on from one generation to the next. So inheritance is the specific one we're going to talk about. And the main man who came up with the modern ideas that we currently understand are associated with genetics is Mendel, and he's the one who came up with the idea of the gene. Before that, we don't have any recorded history that talks about anybody else figuring this out. And we really do appreciate Mendel for his very careful experiments that he did. So let's go on to part one of this lecture where we learn a little bit more about Mendel and his idea of inheritance. And we're going to start out here by thinking about that before Mendel came around, we didn't have a very good idea of how traits were inherited. I mean, maybe some cultures had gotten it figured out, but Western culture, where we have everything written down and we have careful documentation of it, we don't have any documentation before Mendel of this idea being reasoned out. Before Mendel, the most popular idea was this concept of blending hypothesis, where traits blended together, kind of like two buckets of paint would blend together. Like if you blend a blue bucket of paint and a yellow bucket of paint, you're going to get green. And this makes a lot of sense in some ways. I mean, if you think about humans, you have two parents. One has blonde hair, one has brown hair, and maybe the kid has like light brown hair. That kind of makes sense. But when you start really thinking about it, that doesn't always happen. And two people with brown hair can have a kid with darker hair or lighter hair. So how does that work? The blending hypothesis does not really work with that. So Mendel did come up with this idea that we now know as the particulate hypothesis. And the particulate hypothesis simply means that traits are passed along in discrete, which means small and distinct, heritable, that means they are being able to pass on to the next generation, units, and we now call them genes. Although I'm not sure that Mendel called them genes, but that's okay. We're going to call them genes because that's the words we use these days. So Mendel did document his research very carefully through very, very careful experiments with garden peas. And the picture that you see right here is a garden pea. It's the flower. So eventually, Mendel came up with two main laws of inheritance. We call them laws, not because you can't break them, but that's just a word that's used to describe math. And there is a lot of math here in genetics. So laws of math and laws of physics, those are the general principles that are involved. So he took garden peas and he very carefully crossed them together. And I'm going to go through this with you in detail in a little bit. But just an example here, he might have taken a yellow garden pea and a green garden pea. Now, I know a lot of us aren't familiar with yellow peas, but they do come in the variety of yellow. Most of us, when we buy peas, they're the green kind. That's okay. They do come in yellow. So he crossed those, and he went through three distinct generations, which I will go through with you in a later slide. But he always did the same experiment over and over again. So garden peas are great for doing this kind of experiment with because they do have a lot of varieties. And so anything that's like a distinct heritable feature, we use the term character or characteristic. And so we might have the characteristic of seed shape, and you can either have round or wrinkled. Now, the variants of the character are called traits. So the traits would be round and wrinkled, and the character would be seed shape. You could do the same thing for seed color, for characteristic, and yellow and green for trait. Same kind of thing with flower color. Purple and white are the traits. There's also for pod shape inflated and constricted. For pod color, yellow and green. There's also flower position. It could be axial, which notice that the stems come out of near the bottom, the stems of the flowers. Or it could be terminal, the flowers come out at the end of the plant. They could be tall or dwarf. So there's all these different characteristics, and each characteristic has at least 2 traits. So how did he handle this? Well, remember that the flower is the sexual reproducing organ of a plant, and at least a flowering plant. And so, and a lot of those flowers have both male and female parts. So, what he would do would be he would cut off the male parts of the flower, and he would take a paintbrush, and he would take the pollen, and he would dust it onto the female parts of the flower. Now the pollen comes at the end of the stamen, and the female parts, there's an ovary, but there's this whole thing is called the carpel. So male stamen, female carpel. And then he would wait for whatever characteristic to show up. In this case, maybe we're looking at seed color. So there's the seed color right there. Or maybe we're looking at height, or maybe we're looking at flower color. It doesn't matter. We still have to do the same basic experiment where we cut off the male parts and do a very, very careful cross between the male and the female. In this case, he really was looking at flower color, and that's kind of illustrated here by showing you the different flower colors. But again, he did this with every single trait that he could. So if we take a look closely at the series of crosses that he did, and I use that word a lot, the series of crosses, because there's multiple generations that we're going to talk about. The first thing that he had to do was make sure that each variety that he was looking at was true breeding. Such an important concept. What that means is that if you have a yellow pea and you allow it to self-fertilize, which of course is very easy to do in plants, you can self-fertilize them. Oh, and if I just go back up here, I can show you, instead of cutting off those male parts, you literally just let that fertilization happen right there. That's self-fertilization. So if you allow them to self-fertilize, and if you have a true breeding plant, what happens is you get all of the same offspring. The same thing for this green one. This one's also a true breeder. So when it's allowed to self-fertilize, all of its progeny or offspring are all of them are green. There's only 4 shown here, but peas tend to have many offspring, and so 4 is just kind of representative. So let's look at the series of crosses. The first thing that he did, again, was to get these true breeding. So these ones would be true breeding, and these ones would be true breeding. And he would then take and cross one from here and one from here. And again, he would put them in the ground and grow up the flowers, but remembering that this parent made yellow peas and this parent made green peas. And that would be called, if he did that cross between the yellow and the green true breeders, that would be hybridization. He was making a hybrid. And when he did that, he was doing something that he called the pea generation. Now, P stands for parental, not the pea plant. So just kind of be aware, if you're listening and not actually looking at things, that this is called the pea generation, the letter P. P for parental in this case, not P for pea plant. So remember that these are both offspring of true breeders, and they're true breeders themselves. Now they're cross-fertilizing. And when he did this cross-fertilization, he came up with this next generation here, and I'm going to name them just like Mendel did, F1. And it doesn't matter what the F stands for. It means brothers, basically, filial. They're siblings. So all of these are offspring of the parental generation. And 100% of them were yellow. Again, it wasn't exactly 4, but that's okay. 100% of all the offspring between a yellow pea and a green pea were yellow. And he's like, something's going on there. And I'll tell you what that is in a second. But then he didn't stop there. He took two of these, like maybe he took that one and he took that one, and he crossed them together to make the next generation, which is called the F2 generation. The F2 generation. So we could say like this, we could say the parental generation were the parents of the F1 generation. And the F1 generation are the offspring of the parental, but they're also the parents of the F2. When he did this lots and lots and lots of times, about 75% of those peas were yellow, and about 25 of those peas were green. 25% of those peas were green. So he did this with, again, all the different traits. Here you see it happening with the purple flower versus the white flower. The F1 generation were all purple, and the F2 generation were 75% purple and 25% approximately, right? If you do the math here, it's not exactly 3-1, but it's really, really close. 25% of them were white in the F2 generation. So from this, he came up with this idea of the law of segregation. And the law of segregation says that when you are making gametes, you only pass down one of those gametes to the next generation. So that's this idea of a particulate. Right, we talked about the particulate hypothesis of inheritance. So based on this, he also realized a couple of other things. He realized that the factor, whatever it was, which we now call genes, the heritable factor for white flowers was hidden, or we say masked. That's the word I often use in the presence of purple flowers. So we use this term dominant to refer to, and here's the definition. We use the word dominant to refer to any trait that masks the other possible trait in a hybrid, in the F1 generation, right? So the thing that's also important to get aware of this, and we do have a term, recessive, to be aware of. That's the one that is masked. And the other thing to be aware of is that even though the white was masked, it didn't disappear. Like if this were buckets of paint, and if you mixed them together, like if you mixed two purple buckets of paint together, you'd never get a white bucket of paint out of there. It doesn't matter how many hundreds of times you did it. But the particulate version of this hypothesis, that works. If there's a dominant one that masks the recessive one in a hybrid, you're always going to see that. And in the F2 generation, the third generation in this series of crosses, it'll show back up again. So he found the same pattern in all the characteristics that he looked at. He looked at flower color, and this is just a list of which ones are dominant and which ones were recessive. And this is a list of his actual data and the ratios that he came up with. Yellow versus green, round versus wrinkled. Notice that all of them are very close to something that we can think of as 3-1. Rounding to the nearest hundredth place, it's a little bit more or a little bit less than 3-1, but still, those are pretty good data. So the way he developed his model to explain this 3-1 inheritance in the F2 offspring is due to these 4 related concepts, and I'm going to step through them with you one by one. The first thing that he did, and I'm trying to explain this to you in our modern sense of the word, but just please remember that he didn't know anything about DNA, and he didn't know anything about chromosomes. But what he could figure out just from looking at this pattern over and over again, what he realized is that, first thing he realized is that alternative versions of genes account for the variation in inherited characteristics. For example, if you've got your 2 flower colors, you've got purple and you've got white. And we're using the word allele to refer to the different versions of it. So alternative versions of a gene are called alleles. So maybe you have the purple allele and maybe you have the white allele. And each of those are located at a very specific place on the chromosome called a locus. But again, Mendel didn't know about that. What he did know is that there were alternative versions and that they existed both in the purple flowers and in the white flowers. We now know that those are in the DNA and that maybe it's a little mutation there in that DNA, which is causing the change. And of course, that could change the whatever enzyme is made, and that enzyme might lead to making a very particular color of flower. So alternative versions, that's the first one. The second of his ideas here of the 4 is that for each character there are 2 alleles, one from each parent. Both parents are contributing equally to the offspring. Now, Mendel, again, didn't know anything about chromosomes, and he didn't know anything about locuses, or loci is the plural of locus. But he did understand that you could have 2 of the same alleles, or you could have 2 different alleles. And so that's reflected here. Here's true breeders. We now use the word homozygous to refer to 2 of the same. So this one is homozygous, or a true breeder, for yellow, and this one is homozygous, or a true breeder, for green. And we know that when you do this cross, you'd have to get one Y from one parent and one little y, in this case, from the other parent. And so you get the heterozygote. In that heterozygote, right, only one of those colors shows. And so we must know that whatever one shows in the F1, that's the color or the trait that is dominant, in this case, yellow. The other one that didn't show up, in this case, green, that must be the recessive one. I mean, it's got the dominant allele and the recessive allele, right? It's just only showing the dominant allele. Yet the recessive allele has like no effect on the appearance. The 4th point that he came up with, and this was his genius, was the law of segregation. And I referred to this earlier in this lecture. The law of segregation, what this says is that you got these 2 alleles, right? One from each parent, and you have 2 alleles for each heritable characteristic that you have. They separate, or the word is segregate, right? Separate means segregate, and we call it the law of segregation historically. So these 2 alleles have to segregate during gamete formation, and they're going to end up in different gametes. So an egg or a sperm only gets one of the 2 possible alleles that you have. When you make gametes, into each gamete you're only going to put in one of them, one copy. Into the other gametes that you make, you'll put in the other copy. So here's an example of this. You can think about this as chromosome pair 1, and here's chromosome pair 2, and here's chromosome pair 3. And this has the red gene, and this has the blue gene. And this has the light blue, and this is the dark blue. And these ones, they're the same. So when they segregate, this one gets one from the orange chromosome, one from the green chromosome, one from the teal chromosome. And this one gets a different version of it. This one had the darker one, this one had the lighter one. Does that make sense? I hope so. We talked about this when we were talking about meiosis. We called it, what did we call it? We called this, when we were really thinking about this, we called it segregation. We also brought up another idea called the law of independent assortment. But hang on to that. We'll talk about that again a little bit later. So basically, when we're looking at just one characteristic, and we're looking at 2 traits, like purple and white in this case, you end up getting a 3-to-1 ratio in the F2 generation. Now remember, the F1s were they were always hybrids or heterozygotes. So if you take the two possible alleles from an F1 parent and you put them on the outside of this little matrix here called a Punnett square, you can say, oh yeah, there was a big P and a little p, or I guess we had used Y's earlier. We could have said big Y and little y. We always use big Y, big letters for dominant and little for recessive. It doesn't matter really what letter you use, as long as you can tell just by looking at them if it's a dominant allele or a recessive allele. So big letters for dominant and little letters for recessive. So if you put those gametes into a Punnett square and you say, oh, well, what if this sperm meets up with this egg? What kind of offspring would you get? You would get a homozygous dominant. They have two of the same letters. But if this sperm met up with this egg, you get a heterozygous. And you can kind of hear the language I'm using. Well, what if? What if? These Punnett squares are great to predict the results of a cross between individuals when you know their genetic makeup, their genotype. Again, capital letters are dominant and lowercase letters are recessive. And so what you see here is a cross between a big P, little p heterozygote crossed with another one just like that, big P, little p. And these are the gametes here. Notice that they've segregated. We've put them in different cells. Anything with two identical alleles is called homozygous, and I've been using that term a little bit as I've been lecturing, but here's where we get the definition. Homozygous means that they have two of the exact same alleles for each character. So here we go, right here we see big P is homozygous. And then if it's heterozygous, that means they have two different alleles. So here's an example of one with two different alleles. And over here, what about this little p, little p? Well, that's definitely homozygous. But it's different from the big P because the way that they would look. The big P would look purple, but the little p would look white. So we use the word phenotype to describe the way something looks. And I have that on my next slide too, but I have the word here, so let's just go ahead and discuss that. Heterozygous, we just use the word heterozygous. But for a homozygous, we often use the word homozygous dominant or homozygous recessive. So this right down here, we would call this number right here, we call that the phenotypic ratio because it's 3 purple to 1 white. This set of numbers right here, we would refer to as the genotypic ratio because there's one big P, 2 right there, big P, and 1 little p. So we would say we have a 1 to 2 to 1 genotypic ratio and a 3 to 1 phenotypic ratio. The interesting thing here is that if you think about it, the phenotype does not always tell you what the genotype is. In fact, the only time the phenotype tells you what the genotype is with the recessive. You know, if you're showing recessive, you have to be homozygous recessive. If you're showing the dominant, though, you could be either homozygous dominant or you could be heterozygous. And just by looking at the purple, you just don't know. You could be either one of those options. In fact, you got, depending on the population, a higher chance of it being heterozygous than you do of it being homozygous. So since you can't always tell the genetic makeup of an organism that's showing the dominant phenotype, we can use a technique called the test cross in order to figure it out. So let's say you've got a purple plant and you want to know if that purple plant is a true breeder, because all you want to do is grow purple plants. Well, you have to do something called the test cross first. And what you do is you say, well, that plant that that purple plant could be big P, or it could be big P, little p. The sure way to find out is to cross that purple plant with a white plant. Because if you cross it with a white plant, you are going to for sure know whether that is a big P or big P little p purple plant. Again, this is called a test cross, so we're just trying to figure things out. If you cross a big P with a white little p, all of your offspring are going to be heterozygous. And that means that all of your offspring are going to be purple. If you do the same thing, if this purple plant up here is heterozygous, big P, little p, and you cross it with that white one, little p, then when you get offspring, about half of them are going to be purple, and the other half are going to be homozygous for white. So immediately, right away, you can say, they were all purple. My plant must have been homozygous. Or if they're about half and half, you can say, well, my plant must have been heterozygous. Now, we use this technique a lot when we're breeding dogs or cows or horses, things along those lines. This is a technique that farming uses a lot. Well, so you've learned the first basic stuff from Mendel. I hope that you've taken some important information out of this, and we will continue this lecture when we get into this is actually chapter 11, sorry for that typo, part 2.